Hey everyone, I'm Dr. Guidone, and I'm here to tell you a little bit about the research that goes on inside my laboratory. Within my lab, we do a lot of work in the coastal marshlands, and we've studied a number of things out there, mostly with this plant that you're seeing pictured here, Spartina alterniflora, which is within our local marshes, the most abundant plant in the low marsh zone. And we've looked at species interactions between that plant and epiphytic algae, as well as barnacles, a fouling invertebrate organism. We've also looked at how that plant interacts with rainfall. And so how does the plant intercept the rain and how much of the rain actually makes it through the canopy? We've also looked at several anthropogenic impacts, both out in the salt marsh, as well as a few things on campus. And so a couple recent projects have looked at microplastic pollutants, particularly in the salt marsh. And we've also been examining an invasive species that we see on campus. And so I'm going to tell you just a little bit more about each of these current projects. Within the salt marsh, we have examined epiphytic communities. Epiphytes are just plants or algae that grow on top of other plants and algae. And so here, the epiphyte is this algal organism, the seaweed that is growing on top of this Spartina stem. And it may not look like much, it's kind of small, but that epiphytic algal organism is a forest for a small invertebrate animal. And so we have been examining what types of algae grow on these stems, how those communities differ between locations within Georgia, how they differ geographically. So we have collaborators in Connecticut who are doing the same type of survey work, and we're comparing our results to theirs. And then we've also been examining the animals inside the seaweed community to see if the animal community varies with the type of seaweed. And this comes back to our marsh food web. These small animals are an important component of the food web because they're consumed by larger invertebrates and small fish. And so they're an important link between the epiphytic community and the rest of the food web. We've also looked at the interaction between Spartina and barnacles. Barnacles are these small invertebrates that have to settle on a substrate, and in our local habitats, many barnacles end up settling on Spartina itself. And so we've been curious if that has a negative impact on the plant. And we found at most sites, barnacles don't settle in high enough numbers to have a negative impact, at least not something that we've been able to measure. But at those sites closest to the ocean, if you have a marsh system there, those plants get covered in more barnacles. And it seems like they may have a negative growth impact that they experience. We've also looked at how these plants intercept rainfall. And so we actually have a rainfall simulator on campus. That's this middle picture on the bottom. At the top, we have some sprinkler heads that release water that in such a way that it falls like rainfall. And then we measure how the rainfall flows down the stems by funneling some of the stems, and we can collect that water in bottles. And we also measure how the rainfall falls through the plant canopy with these little cups. And this first picture here is actually looking up through the stem canopy so we can get an idea of what type of coverage is occurring over those gaps in between our pots. And this is a collaborative project with Dr. Van Stan. And ultimately, we want to understand both how the plants are interacting with the rainfall, but also how that might impact coastal marsh erosion, because that is a huge problem for many coastal locations where salt marshes are the habitat. We've also looked at anthropogenic impacts. That just means human impacts on the environment. And one thing we've studied a lot is microplastic pollution. And we've looked at the microplastics within the sediment out at the beaches, as well as the microplastics within the algal epiphytes that we've been studying on Spartina. And it does seem like those algal epiphytes are trapping microplastics, and how much microplastic they trap depends on the type of algae, so what species it is. Lastly here, on campus, we've been looking at an invasive species of insect that impacts magnolia trees. So on this leaf on the bottom, all these little white dots, that is the invasive species false oleander scale. And it's a type of insect that is a phloem feeder. It feeds on the sugars that are inside the veins that the plants have produced. And we're specifically interested in whether or not infestations of these trees is impacted by where the tree is planted. And so we're currently comparing infestation levels of plants that are located in the middle of campus, so they've been purposefully planted as a landscape tree, and comparing that to plants that are found in more natural settings where they haven't been purposefully planted. And so that's a little bit of what goes on inside my lab. If you guys have questions, please get those to Dr. Sargent, and I'll be answering those in the future.